this visible light actually is only occupied a very limit like wavelengths, like spectrums of the whole like those photons. So it's around between like the wavelengths, it's about like 370 land to like 750, and that's the visible light. And below this, like shorter than this, then that's the ultraviolet. And longer than this, that's the far red or infrared. For all the other for rhodopsins, for like human eyes, or from the animal, or from the microbians, those rhodopsins, so they sense like the violet light, the blue light, and far the most far is like around 750, and it's really very hard to go even further. But like a Richard and like the Roger Chen's lab, that's so hard. They try to like the red shift chromosome, and it's really hard to achieve and that goes. But like, but today we're talking about another groups of the photosensors, and that's the phytochrome and the CBCRs. See, it looks like by nature endogenously they're sensing the red light. So I think this is the most important property of this it's a photosensors that catch like attention of many scientists, especially for in the field of optogenetics. I'm a little worried. Okay, now let's first take a look at the four purine structure. Let me see, I should put, yeah, it's from the ham, it's from, so this pigment is, and it's like coming from the ham, and that's the beading, it's called a beading pigment. So it forms the color for like the red blood cells, those red color, because they're binding on an iron and give this a red color. But if the whole the big rings is separated, then the bilivendin, then it will sense like it will give a green light and some little structure change. Let me see, between these two, not a lot of structure change. Oh, here we can see it's a here, then shift to, it's like it forms like a hydrogen bond, and some involved in like a molecular interaction, and it will cause some difference for the most sensitive their light wavelengths, and some little more structure change, and it will induce like to the dark blue or even purple. And after like some enzymes digest, and it will go to this group, and they will sense like it will have a yellow color. So let's go back to see to zoom in to see the linear texture skeleton. And the parent have a 4 parent A, B, so each one labeled with A, B, C, and D. And so majority those interactions with the proteins are at this side. It's labeled like a 3 to 1 or 3 to 2, and that's usually that's forms the chemical bond with those photosensors. And another is the carbon 10. and sometimes in some of these like a phenochrome for these light sensors, some also form like a connections with this carbon 10. So actually today I'm going to, yeah, and today I'm going to like, okay, so still give me some pointer, highlighter. Okay, so this BV is the precursor of the various building pigment. So all these groups, this pigment, and the precursor is the BV. So the chromophore is also called the BVs. So for this, P5B, PCB are enzymatic biosynthesized from BV in cyanobacterial, algae, and plant. So for those like the photosensors from the cyanobacteria, algae, or plant, and these chromophones, this we later will call them FIS and the CBCR. It can alter catalytically form a covalent bond with the chromophone, and their chromophone is the BV or the BV families. For this PVB, so then for the PVBs about synthesized from PCB, then alter isomerize activity of certain CBCRs. The in vitro experiment shows that the biliverdin reductase, the BV reductase, can carbonize the BV P5B PCBs, those that was being blocked by this chat box, and those with sense like blue, dark blue, and purple ones, and that can carbonize into these ones, and respectively as chromophone inactive form for the photoreceptors. So it will be reduced from, so this will be reduced with some enzyme and from this that is marked with yellow one, and the yellow the yellow color, that means that they're in chromophone inactive conditions for these chromophones. So again, those are the very canonical losses, those with the GAF domains. It will bind in the 3 to 1 or 3 to 2. And those for the PAS domains, some CBCRs have the PAS domains, and they will bind into this the 3 to 1 site form the chemical bond. And BV have double bond between C31, C32s. And for some other CBCRs, there's also binding to form bond with this carbon number 10. There's a very specific, how should I say, like a structural property for this FIS is like, it's called like a town structure, and also a node structure. So in image you see it's like to indicate the node structure, and this to indicate a town structure. So these interactions will cause like a conformation change, and that's what's being induced by light. So that's how the light trigger the whole conformation change of the overall structures. And the CBCRs, like XRG or the XCF, they're like kind of like have endogenous like the cis residues. Then have not only the kind of cis but also second cis in the DXCF motif, and second cis attached to the C10 of the chromosome contributed to a larger spectrum shift. The DXCIPGAF domain initially incorporate PCB and converted into unknown chromophore PXB, and that's CBCR. Let's go on to look at the tertiary structure of those two photosensor. One's the Phi and one's the CBCR. One can see for the Phis, usually they have many like domains, like a PAS domain, GF domain, and a Phis. And there's a tongue that's like a 2 beta sheet that's insert of go further, then have some kinds of like interaction with the node that is from the PAS domain. But for the CBCRs, the binding site chromosome is in the pocket, the GF. And for the Phis, they also have the same domains that called the GAF, and the chromosome binding pocket is around here. So for the two that like its neighbors, the PAS Phis is likely have some like interaction, and that will have involved in the total function of these Phis photosensors. And here are some examples like how those chromophores are inserted into these binding pockets. So this shows another diagram. It's a topological structure of the PCMs, the Phis and the CBCR. And PCM means the photo core module that sends the photon. So the tom structure that goes inserted into here, and the binding into the node structure of the PS. So this and is the absorption result for each photosensors, like one by one, like that's okay, they are matched. So it shows that for PR and the PFR, like different forms, and it shows different most response wavelengths. So that like the representative, the Phis and CBCR shows variant photo conversion between the two absorption forms, red or far red, D13 PFR or PR, or the blue-green, D4 at PB or PG. Let me see, PB or PG, blue and green. Yeah, it's here. This one PB and the PG. So one form that's in absorption, that's the blue light. Another form is the green light. So it shows that there's a two absorption form, and for each forms for each conformation and its most absorption, the wavelengths were different. So between these two forms, like one form is like we can say that it's an active form, another form is an rest form or dark form, something like that. So different forms have the different wavelengths preference. Then let's to focus on the forms of the tongue and node structure. The node is formed by a strap structure of N-terminal region of the PAS, So here the blue one are not PAS domain and passing through a loop of the GAF domain. So the loop is provided by the GAF, and green one, the loop. And the blue one is the node that's coming from the PAS domain. And the structure between the PR form and the loop and node between the PR form and the PFR form is that here is a beta sheet. However, for the PRF form, the far right absorption forms and the loops is changed to it's an alpha helix. So okay, here. So this is the closed state and this is the open state. So here we know that it's kind of like the conformation is quite dramatically, right? So the tongue, which is the extended region from the fifth domain to neighbor of the chromosome. Oh, that's another important thing. So we already know that those phorodopsins they prefer to form a dimer, and so as for these phase and the CB phase, no phase, not the CBCR. So here we're only talking about the phase. So these two actually forms is a dimer, and they formed but they're closer together. It's so as neighbors. The binding pocket of the GAF domain is involved in conformation change, involved light absorption. So this is the tongue structure, and it's important for light structures. So here we go further to take a look at the tongue structure in PR form, and it's a beta sheet, and this distance between this the loop and this yellow structure, and that is the that is the no okay GFPAS phi GFPAS phi. Yes, it's called phi. Although like okay, it's of PR forms, and this two distance is longer. However, but after the tongue structure changed to a beta to alpha helix into the PFR forms, and the two distance becomes shorter by like larger than the 2A's. And here this shows a whole family of these CBCR GF domains. There's quite different between different CBCRs, and it shows some examples of or list the different CBCR GF domains. So for the photo conversion mechanism of the CBCRs, and this important process named it's a flip and rotate process. And the flip is at this chemical bond, and after flip and the whole structure and the pyrrolines is rotated into another structure. So this conformation change is the critical for the photo conversions. So for the PB form and PG form is flipped here and rotate, and then from the structure from change from here and to here. And this structure change will change the whole like the distance, and there's a more linear becomes like more circular like the structures in the world all change the absorption the wavelengths. So shining with the blue light and will change from this form to another forms. and like shedding with green light or red light, it will cause like some the proton will shift from one place to another place, and this shift will cause the photo absorption and light wavelengths change, and also blue light and the sunlight, and this conformation change is called the trapped twist mode. So there's a like a flip at this area. I can see this flip, and this flip will directly cause that forever as those pi like the electrical cloud is covered this area, but now it's only covered this. So the sensor light wavelengths were changed. So for this chapter, I provide example of optogenetic tools card. When you zoom in, we can see that first, for example, and the PFB chromophores, and it's being combined with East2 hybrid system and cause like protein like interaction that is induced by light. So now let's talking about something about the physiological function of the FIS and CBCRs. First, FIS and CBCRs are known to function as a kind of light activated trigger, which alter a biological behavior. So, for example, the FIS interact with phytochromo interaction factor PIFs, which serve as a transcription factor. It can regulate the gene expression. So that's one function of those light sensor FIS and CBCRs within a biological behavior. So this shows like here, like when shining with the green light, and it becomes active state and it has a autokinase like phosphorylation itself. Then for the phosphorylation, it's a receptor, and after phosphorylation, it becomes an active transcription factor that can turn on the gene expression. So it's quite similar. It's another example as shown in here. So also it's a function as a like a sensor light, because like for this cyanobacterias, it can sense like in the white light, like the green blue light, and also in a far red light condition. But these are two photosynthesized conditions. They're using different like a machinery. So like usually like so function when it's in a white light and the white light conditions, and this were harbored accumulation of only SH1. So it were harbored this SH1F. So then under the far red light conditions, and so this harboring this inhibition effect is being released, and this FIS is not activated. So this photosynthesis are being activated. So this guarantee that there's efficient photosynthesis even under far red light conditions. Another structure, like in the focal sums, is optimization of light harvest intervals. So it really forms like a complex and a small molecular device that formed for such a forms such complex here, and it functions in this the synchronous PCCP. Looks like a green algae, but it really like forms like a communities here. So there's several like the single cell those microbials, but they form together becomes a very large communities. That's and the scale is around in millimeters. So here that can be visible seen by eyes, and but each dot here at the form as is a single cells. So it's not just a diagram; it's really like very close to the true structure, like the photos of this. This is a EM image, show that the real structure is really look like this. Looks like a bunch of flowers and like planting here on this luminant surface membranes. And to take a look at for the structure of these fingerbelisms, and so each part is kind of like one like composition that insert that forms a complex here, and each one will have a chromophores are binding, and it has so this whole machinery is from like a concentration of the energy synthesized complex. So for this multi-cells, those microbial community, and also this light sense input system that induced by CBCRs are also involved the positions of those single single cells the location within the community. So for those deep inside, like in the center of this of the whole like whole groups of cell communities, and this is C di GMP is off, and there's only green light can penetrate here. So when it brings it penetrate here, so it will cause these cells to go up, go further, go to the outside, go to the surface, and after when they go to the surface, so this green part they go to the top, right? And so then when here, so there's nothing change for the center one, because center one they both can capture blue light and green light. However, for these cells, there's no blue light furthermore, so this CD GMP has been turned off. Then it becomes a green cells. So these green cells without the blue light stimulation, and it will cause them to move back again to go to the surface of these microbial communities again. So the locations of each cells is really determined by the light, how much light can penetrate into the whole like groups bundles of those cells. So it's really like so, oh, I need some light and go to surface, and surface there's blue light, there's green light. I'm pretty happy. I'm happy now, and I turn on this C9 GMs here. Then later I cannot. Then I go deep, because some others they go up, go to the surface, then go to the center. I feel that oh, there's no my the blue light signals. Then I must like move up, go back to the surface. It's a quite interesting. So we can see here that the light can really like modulate like the behavior even in this cyanobacterias. It's quite like the performance, like the plant, right? If there's not enough light, and some trees like let's go up, and if there's no other big trees around, there's only grass, so they may not need to go so high to capture like the light. signal. So light is so both ways. So for this specific process, we call that the light is a signal. But I think like for that, but the reason is that the light requires the light to to continue for their survivals because they need the light, like a plant need light to do the photosynthesis to generate the materials for their survivals, for their lives. So this image diagram, I like it very much because it summarizes a lot of optogenetic tools, of course, including our superstar today is the phytochrome form-based system, and showing here that after shedding the light, it forms the two components like binding together. It also forms the interaction. Of course, there's other optogenetic tools like the channels and like the killers and other like histone kinetics. And so for really, I'm quite interested with this binding process because it means that so when shedding the light, so this connection, protein interactions that can be controlled by the light. So we know that a lot of signal pathways require that, like some signal like an inactivation require those proteins to come together and the signal turning on like a kinase and some signal pathway cascade require the cells the complex to dissociate and release some part like the beta catenanes to induce gene expressions. So I think like especially like for the gene expressions and the low signal cascade, I'm thinking that this Phytochrome-based system could be very useful. But oh, okay, so this is already like successful in the zebrafish systems. So it's summarized here. Within the light, it causes like a protein interaction. Also here and here, and some cause like the light stimulation will cause the proteins to separate, and like for this example, like cause those in the cytosol like bind come together then and forms a complex interaction with each other after with the light stimulations. Okay, so we already go through this newly there's a pigment BV and the molecular sensor that is using this as a chromophore, as a fees and the CBCRs. And here is the reference we're citing today. So still, today lecture, we're going to have a quote that light is one of the most important environmental signal, not only for photosynthesized organism, but also for non-photosynthesized organism. So it's not allowed, not just like some animals that have best visions, like eagles, like owls, those have so wonderful visions. Also for like a human being, we can see like a so colorful world or a butterfly that have like a six photopigment that can see a so colorful world. Then not only important, light is not only important for those organisms, but for those cyanobacterias, we know that light can really, it's kind of important a signal to change their behaviors in a dramatically uh like areas and different functions like circadians like a feeding and like a breeding and a lot of things and also some like how they forms a society, how they separate, how they like change the position of the flagellation. It all can be controlled by light, and this is not engineered. It's naturally happened. It's quite cool, and thank you everyone. Thank you.